about a couple other intracellular pathogens like Listeria. We've talked about like chlamydia, some interinvasive E. coli that cause lots of membrane ruffling and disturbances to the cell upon entry. But if you follow along this cartoon at the bottom here, it kind of shows how these members of the P-complex at the phylum enter cells. So they attach via that conoid receptor on one end of the cell. They form a pretty tight junction there. And then this junction becomes what's called a moving junction. So it's almost like a hand going into a glove. It forms a tight seal that does, again, like very minimal damage to that host cell. And then the whole parasite slowly just slides into its own little endosome. And then that moving junction seals back up when it's done entering. So because it's able to do that, it doesn't trip off a lot of these pattern recognition receptors inside the cell. There's minimal attention from that host cell as it's being invaded, not a lot of damage, and it gets in its own little vesicle that way. All right, so getting into malaria a little bit, these are recent data on the global burden, the number of cases worldwide, about 263 million cases a year. This number does tend to go down over time, so this is pretty good. WHO and a lot of other agencies have made a lot of efforts to try and control the spread of malaria. That said, there's still a huge number of deaths per year, about 600,000 deaths every single year to malaria, and the majority of those deaths, about 75%, occur in children under the age of 5. We talked about before the fact that they don't have highly developed immune systems. So there's a little bit of protection up to around 4, 5, 6 months of age. So the babies still have a little bit of maternal antibodies if their mother's infected. But by and large, kids are just not able to fight off infections very well. In terms of transmission, by far the most common mode of transmission is the bite of mosquitoes, in particular this one genus of mosquitoes called Anopheles mosquitoes. They have a wide global distribution. Other forms of transmission include blood transfusions, which is very rare, injection drug use where you mainline parasites into your bloodstream. And this parasite can also cross the placental barrier as well, so you can get congenital transmission. In the US, we do have some cases, but most of ours are imported. We had a case of one of these species called Plasmodium vivax back in 2003, and then we had another case, Octoctonus, so acquired here in the United States, not imported. We had another case last year. I'm not aware of any cases this year that have occurred during Octoctonus spread. But import cases, yeah, we get plenty of those. So, like when I worked at Madigan, we had a couple different times where we had test orders for soldiers who were suspected of having malaria just based on travel, import cases, things like that. These 5 species are current species that are human adapted. Molessi, this last one we'll talk about, but this is a zoonotic one where the main reservoir are macaques, but the rest are all mainly human reservoir species of Plasmodium. Falciparum is one of the most deadly of these 5 species. If you look at the range of Falciparum, range covers a lot of Africa, and that's indeed where unfortunately most of the deaths occur worldwide. 95% of deaths occurring in Africa. Vivax, we'll talk about that distribution too separately, but that is probably the widest geographic distribution. So we get cases of Vivax in Central America, South America, deep into Western Pacific Islands, throughout Africa. Lots of co-infection areas, lots of co-endemic areas. Ovale has a lot of a similar geographic distribution as Vivax. Malaria has the most restricted, or one of the most restricted ranges. So you put them together and you do have a lot of global threat, a lot of exposed vulnerable populations. Yeah?
With the malaria, the map, most has this issue. I have a different one. Is the map that we're showing up here pertinent? Something we need to know the geographic regions?
No, we'll go over all of those, we'll go over 2 of them separately, the 2 that matter. And you should, if you have the PowerPoint, you should have these separate ones in there as well. But you don't need to know them. It's just kind of like there's a narrative here. So most of you guys have the PDF or do you have the PowerPoint? Mixed bag, it sounds like. Okay, well, it doesn't really matter. Most of this is just for atmospherics and background. But when we get to SIPRIM and buybacks, we'll definitely look at the maps there for those two. So historically, the point this map is making, which you can see information from these maps, is that the ranges of malarial regions has changed a lot, just in a short period of about 100 years. In the United States, we used to have a lot of cases of malaria, autochthonous malaria acquired here, like going up into Canada in some cases here. And that went into the 1900s. So much broader ranges. USSR used to have severe malarial problems as well, places you no longer necessarily think of as regions. That complicated setting up our nation's capital in DC. It was also the reason, if you ever wondered why the CDC was placed in Atlanta in the 1940s. It was because that was like the epicenter for where malaria was in this country. So they set it up in Atlanta for that reason. And then our eradication efforts were pretty successful, and we've driven out most of our endemic malaria since then. One more map. This kind of shows you that even though you have a lot of widespread regions for getting malaria, different types of malaria from different species, most of the cases are concentrated here in Africa. And a lot of those are, unfortunately, the most deadly, Plasmodium falciparum. The reasons for this are probably mostly attributed to the fact that you have this particular species, or complex of species from Anopheles called Anopheles gambiae. Those mosquitoes tend to be super anthropophilic, meaning they like to bite people specifically. They're very aggressive, and they're very good at transmitting the disease. And they live a long time too. They live longer than other mosquito species, so they're capable of transmitting a little more effectively. In terms of the life cycle, you get that mosquito bite most of the time, and then transmission again. And in the salivary glands of the mosquito, they will inject an infected form called the sporozoite. The sporozoites get taken up into circulation and they go to the liver, which is a very thirsty organ in terms of blood supply. So they'll go down to the liver, and there they're able to bind specifically and infect and enter hepatocytes. In the hepatocytes, you have this first round of reproduction and amplification of these parasites in the human body. This particular cycle is called the exoerythrocytic or liver stage of malaria infection. It's usually fairly short-lived. Most of these species, it takes a little over a week to get through this liver cycle, where they're coming in and infecting hepatocytes as sporozoites. But when they leave, they emerge as a different form called neurozoite. Okay. Plasmodium malariae has the longest incubation cycle of any of these. That takes about 2 to 3 weeks, but most of them are much shorter than that. Once these parasites emerge from those liver cells, they come out as merozoites, like I said, and they come out in huge numbers. So while you might initially get infected with maybe a couple dozen sporozoites from the bite of the mosquito, once you get done with this liver stage, you have thousands of merozoites that are infected, that are released into the blood supply, and there they start targeting erythrocytes. So this is the beginning of the erythrocytic cycle. This is when those infected merozoites will enter those cells in that mechanism that I described earlier and start feeding off of the hemoglobin inside of red blood cells and developing from these different stages that are here on the screen that we'll talk about in more detail. Start out as a ring stage, then trophozoite, schizont, and then schizont is the last stage that they go through before those red blood cells become two-packed and they simply burst. They burst open and release tons of more infected merozoites. So this cycle kind of continues, and it is called a cycle. So you're cycling from one cell to another, and this continues. And that is the disease of malaria. There's a lot of pathology associated with the cycling of invasion of erythrocytes, feeding off of them, and then rupturing them. A small number of those infected blood cells have parasites that start developing as differentiated, specialized gametocytes. Like between 5 to 10% maybe become specialized gametocytes. Most will continue cycling through the stage sexual reproduction. But the ones who differentiate into gametocytes will go into circulating through the blood anyway. But they're the ones that are infected for mosquitoes. So when a mosquito bites a person and they pick up gametocytes, that's what allows the parasitic cycle to continue. And in the mosquito host, those gametocytes will fuse, there will be sexual reproduction, and you'll get continuance of that infectious cycle. So if the mosquitoes are the host where sexual reproduction occurs, what does that make them? The definitive host or the intermediate? The definitive host, that's right. So mosquitoes are the definitive host for malaria. All right, talking a little bit more about the pathology of malaria and how that tracks with what's happening in that erythrocytic cycle that I was mentioning. So when you have a bunch of affected blood cells, a couple things are happening there. First, you have these very hungry parasites that are a lot of the glucose in the cell. They're also breaking down hemoglobin, metabolizing the hemoglobin, using the amino acids from the broken down hemoglobin for their own synthesis and replication. So that's taxing on the red blood cells, and that contributes to a little bit of the early pathology, a little bit of the anemia, lowered oxygen carrying capacity of red blood cells. But what really contributes to the characteristics of malarial disease is when you have a mass rupture of red blood cells. When that happens, not only do you have the merozoites themselves, the parasites, coming out into the bloodstream, ready to infect new red blood cells, but you also have toxic byproducts like hemazoin, which is a malarial pigment that gets released. You have lots of cytokines from the host that get released. And all of that contributes to like one hell of a fever. So you spike a fever, usually coinciding with a period where the erythrocytes are rupturing en masse in a synchronized way. After you clear out the pathogens, your spleen will start clearing out those pathogens, your spleen will start clearing out some of those damaged red blood cells, and your body will break down some of those malarial pigments. After all that fever will subside, and you will have a period where you're more or less readjusting and normalizing. But as you're stabilizing, your hypothalamus is still having to adjust and compensate for that huge fever that you just spiked. So when it does that, you get cold symptoms. The next slide will also go into this and discuss it a little bit. Those cold symptoms are when you have vasoconstriction, so blood is trying to go protect your internal organs. Thermostat is set to a point that's probably inappropriate, and you get severe chills and shaking of rigors at that point. So following a fever, you get chills and rigor as your body recovers from that infection. And then you get a little bit of a lull where at that time there might not be a lot of pathology, but those released merozoites are infecting new red blood cells. So that cycle is going to continue once those new red blood cells rupture. So all that is to say that depending on the type of species of malaria that you're infected with, you might have a different fever pattern that emerges. That's called the chromaticity. That's very distinctive for these different species, but not always followed strictly. For falciparum, initially you get this quotidian fever pattern. This is based on the Roman numbering of days. So every second day, day 1, day 2, you're spiking that fever. It peaks. You get infected on Monday, it means your next going to be Tuesday, Wednesday, et cetera, et cetera. Another species called Knowlesi also adheres to that quotidian periodicity. Vivax and Ovale adheres to more of a tertian fever pattern, which means you spike fever on every third day, 1, 2, 3, just like that, 1, 2, 3, 1, 3. And then the quartan fever pattern is associated with Plasmodium malariae, which is that one that has a long incubation period, a long infectious cycle. So every fourth day, every 72 hours. In terms of just generalized signs and symptoms of malaria, flu-like symptoms initially, when you have initial invasion of red blood cells. But then eventually, even if they're not initially synchronized, they will fall into a synchronized pattern and you'll settle into this paroxysmal cycle. At the same time, some of States, other symptoms are also common with malaria cases. Vomiting, diarrhea, nausea, fatigue, extreme fatigue. Your body is simply wiped out from spiking fevers, from therapy infection. It's just exhausting. This is a disease that I got when I was in Canada, so I can tell you firsthand how miserable this is. I remember I was living up in the Highlands, which is not really considered an endemic area. This is chilly up there in the Highlands of Crecho. But travel all around the country into malarial regions, and then everybody was going off prophylaxis. There was like 10 of us out there from Walter Reed, most in Nairobi, some in Kisumu. But like, if you start off prophylaxis, everybody just drops it after a while because it's hard to sustain long term. So it's not prophylaxis. Picked it up. Was incubating it while I was returning to the United States. I'd been there for about a year at that point. I was returning to go to a tropical medicine conference around in New Orleans. So I was in a Class A uniform and I was feeling bad, but I attributed it to having just flown across the world from Nairobi. And I was walking along past Canal Street from the site of this conference back to my hotel room. And I remember just all of a sudden vomiting, like into a palm tree on the sidewalk in public in classes. So a rapid onset. So I came back to my hotel room and like, probably a fever cycle at that point, and started experiencing rapid rash I've never experienced. You've never experienced what it's like to have those cold symptoms. It's horrible. I think that was the worst part of it for me. It was worse than the fever. But I could not get warm and I was shaking violently. I turned the thermostat up in the hotel room all the way as high as it would go. Got into a super hot bath and was still just shaking uncontrollably. And I stayed there in that hotel room for that entire 5-day conference, just miserable and sick, and hoping I didn't have Ebola. And then had to get on another flight and go to DC, see you at Rare for like my next job, and then go down to Charlottesville. I took a bus, a Greyhound, from DC down to Charlottesville. And it was just, oh man, I was like barely lucid when I was interviewing for my grad school position. So yeah, I mean, I have a strong connection to respiratory disease. I have not get a lot of diseases, but I can tell you it's just like a miserable disease. And that fatigue is awful too. We were just wiped out. Yeah, and then when I got back, I was telling my study coordinator, Margaret, about it. She was like, oh yeah, that's normal. Malaria is so common in those areas. And if you are repeatedly reinfected, you do get considerable immunity to it. So most adults have pretty mild flu-like symptoms in those endemic regions. And they tend to kind of trivialize it a little bit, but it can still be deadly. And then when you come back to a non-endemic area, you have immunity for about a year, but it's not a lifelong immunity. So, unfortunately. So anyway, back to this. Children are a little bit trickier to diagnose. Some of their symptoms are super non-specific. When you are talking to infectious disease docs who work in the tropics, malaria is always up there in the differential diagnosis. They're always considering malaria because these non-specific symptoms. I mean, yeah, you have a periodicity, but you may not be able to follow that. So basically, when patients are presenting with a fever, or febrile, high fever, malaria is always going to be on that differential. In endemic regions, you might not get any sort of lab testing at all because they kind of just assume it's going to be malaria, especially in hyper-endemic areas. And with import cases where the index of suspicion for malaria is very lower, like in that case in Florida that happened last year, probably low index of suspicion there. But that too is easily mistaken for some other more common thing like influenza, just depending on perhaps it's in the middle of influenza season, who knows. So lots of diagnostic challenges for malaria. We'll talk about how we diagnose it specifically in a couple slides. That last slide was discussing some of the common signs and symptoms of malaria. Occasionally, you can get severe, very life-threatening forms of malaria too. So they come with their own specific symptoms, which are super. About 90% of these cases that would qualify as severe or complicated malaria are caused by Plasmodium falciparum. by far, like I said, the most deadly. And there are a couple features of Plasmodium falciparum that contribute to that enhanced virulence, if you will. So one of them is their ability to, when they infect erythrocytes, they cause those erythrocytes to express these very sticky adhesion proteins on the surface of them, and that causes cytoadherence. So when the cells are going through circulation, when they're going through the vasculature throughout the body, what will happen is they will stick to endothelial cells and cause obstruction in the microvasculature throughout the body. That is responsible ultimately for a lot of these symptoms, at least indirectly, that we'll talk about, but that's cytoadherence and obstruction of the microvasculature. What also happens when you have Plasmodium falciparum infections is, since a lot of these erythrocytes are sticking in the microvasculature, it means that they're less likely to be found when you try to diagnose that patient. So you try to take a blood specimen, you don't see any parasites, even though the parasitemia is high, very high. Find is over 5% of erythrocytes infected. And that's because they're sequestered or they're taken away in the microvasculature throughout the body. So it can be tricky to diagnose for that reason as well. When the blood vessels that serve a lot of your vital organs, like your brain, liver, kidneys, start getting obstructed with parasites through cytoadherence, you get kind of like a pile-on effect with impaired perfusion oxygen delivery to those tissues, so they become increasingly hypoxic. That causes a lot of dysfunction. You also have a lot of immune system attention because they're lysing those red blood cells too in your roots. So you get a lot of cytokine release, you get a lot of release of pyrogens, a lot of release of toxic byproducts. As I mentioned, those parasites themselves, especially when you have high levels of parasitemia, a lot of red blood cells are infected, they're metabolizing a lot of glucose, so you get hypoglycemia, extremely severe anemia because your red blood cells are being depleted in huge numbers, so your oxygen carrying capacity is reduced. All of that can contribute to some of these really fatal organ-specific effects with ARDS, acute respiratory distress syndrome. You start again like having your ability to oxygen blood compromised, metabolic acidosis. This will kill someone when your blood pH starts dropping. You have a buildup of byproducts, and you just cannot clean out those toxic byproducts throughout the blood. With the kidneys, there's a nickname for malaria called black fever, and that's due to the fact that you have a lot of pressure put on your kidneys to now clear out all of the damaged erythrocytes. Even uninfected erythrocytes can be filtered out, and as a result, you get blood into the kidneys. The kidneys themselves have to filter out hemolyzed red blood cells, and that results in sort of a tinged urine, which lends itself to the name black water fever. But it's this inky black, sometimes called like tea-colored or coal-colored urine. If you start diluting out that urine, you can start to see that blood, like more reddish tinged blood. But that's a very poor prognosis for patient survival. It means your kidneys are basically shot at that point. We've talked about disseminated intravascular vasculopathy before, other body disorders with other diseases. This too can start contributing to cardiovascular collapse, formation of myocarditis throughout the vessels in the body. So, kind of all that. Oh, and cerebral malaria is probably the most important one. This is the #1 cause of death in kids when they get cerebral malaria. And that is when the blood vessels in the brain basically are obstructed, and you have the brain itself is hyper-perfused, so not oxygen to the brain. So definitely a medical emergency. There are lots of treatments. There are lots of antimalarials that a patient can be on, but supportive care, making sure that patient has oxygen, making sure the blood pressure is maintained, like basic life-supporting care is important with severe and complicated malaria. This slide is just an FYI for you guys, something to consider. But I've been asked in the past how to father, where we spent a lot of time talking about these different species and how to differentiate them through thin and thick smears. But these are some good compelling reasons why it's important with Vivax Valley in particular, I had not mentioned this, and I will mention it again. But with a unique feature of both of these particular species is their ability to form dormant infections in the liver. So it's extremely difficult to treat infections caused by those two species. Specialized drugs like primaquine to treat quiescent dormant liver disease caused by Vivax Valley. So that's important. But the drugs that are used to treat Vivax Valley induce a lot of oxidative stress on the blood cells. So if you have a disorder of deficiency in this particular factor called G6PD, glucose 6-phosphate dehydrogenase, the enzyme that helps you detoxify toxic oxygen species in the red blood cells. If you have that disorder, then treating somebody with primaquine or tenaquine can cause like a massive hemolysis of the blood cells. So if you've ever wondered why the military does G6PD testing, for that reason. So we send people into malarial zones, and we need to know whether or not we can treat them with drugs that can target these particular species of malaria. The prognosis does vary by species, so if you are able to tell definitively that somebody has falciparum versus Vivax or Valley or malaria, that would be helpful, that would be beneficial. Knowlesi is also causing high levels of parasitemia and hyperparasitemia and severe disease. That would be another one that would be important. Skip public health importance, but going down to diagnostic accuracy, the ability to detect co-infections is also important because that would have implications for treatment as well. Many of those regions of the planet are endemic for multiple species of malaria, so be able to identify multiple species. And then lastly, unfortunately, we do have significant resistance to different antimalarial drugs. So in areas where it's known there are a lot of circulating strains of Plasmodium species that have antimalarial drug resistance, we have to alter our treatment regimens accordingly. All right, malaria diagnostics. We talked a little bit about thin and thick smears before. You're taking a blood specimen from the patient, you're putting it on a slide, you're going to drop the blood for a thick smear, and then feathering it out on the other side for a thin smear so you can spread out the red blood cells and take a really look. You usually start looking at the thick smear first. Think of that almost like a screening test. Very high sensitivity relative to the thin smear on that slide. If that patient does have Plasmodium parasites in their blood, you're most likely to find it in that thick smear. So you put it on oil immersion, Radax magnification, look at that slide, check out these good fields under that magnification. It takes a long time to scan this slide before you call something negative. Okay. If you do find evidence, something that looks like it's malaria, that gives us parasites with the same this purple color. If you do find something that looks like a parasite, you can go to the thin slide or the thin smear portion, that feathered out edge of that thin smear, and start looking to see if you can look at the morphology of the parasites themselves. That thin smear is also useful for quantification. I mentioned that hyperparasitemia is defined as over 5% of red blood cells being infected. So you need to quantify based off of the thin smear what the relative or approximate level of parasitemia is. Similar to what we've talked about with other parasitic infections, a negative slide, even after scanning extensively 100 different fields, doesn't necessarily rule out malaria, especially if they continue to have the symptoms. So you continue to take blood every 12 to 24 hours for up to 3 sets before you call it negative at that point. And hopefully you do have one of these adjunct tests available to help you with that diagnosis. For patients who are diagnosed with malaria, you can monitor the level of parasitemia after you put them on some treatment, ACTs or another treatment like that. See if it's having an effect in reducing the number of parasites in their red blood cells. These adjunct tests, I have a picture of the Binax Now rapid diagnostic 4 different species of Plasmodium. Falciparum, Vivax, valley, and malariae. Doesn't do no less than something. It's a good test. It's very rapid. You get an answer very quickly. It's very cheap and affordable. Places to do that. However, the specificity varies and the sensitivity varies as to which species you're testing for. It's very good at testing. Plasmodium falciparum, pretty bad at Vivax, worse at Ovale, and then absolute crap when it comes to identifying Plasmodium malaria. And it's useless for policy. So kind of diminishing returns for that rapid diagnostic test, unfortunately. But good for falciparum. Okay. PCR is super sensitive and super specific. If you want to speciate, you can do that very easily with PCR. The problem there, of course, is just one of access and resources. To have PCR available, you have to text. In a lot of these resource-limited settings, the answer is usually no. But if you do, not only can you speciate, you can also do quantitative PCR as well and monitor for the production of parasitemia. Alright, this is just a composite slide looking at what these different species would look like in blood smear. Main point is you really can't differentiate them very well based on the big smear. Basically just saying the parasite is present in those red blood cells. For falciparum, notice 2 of those stages are not there, the toxoids and schizons. Any guesses as to why that is? Sequestered, probably. So you're less likely to find toxoids and schizons because that is probably sequestered somewhere in the microvasculature. Okay, here we are. Thin blood smear. Very busy slide, but this is a slide to study off of. On the right, this text, it sort of talks you through some unique features to look for, but we're going to go through all of those in some detail here. But notice, as we're looking at these 5 different species, including the Lessii, and these 4 different life stages that are distinctive for identification purposes, how different all of these are. Or maybe they look the same to you at this point, but hopefully after we get through this portion of the lecture, they start seeming different enough to distinguish them. Okay, so initially, when that, are you guys doing okay? It looks like everybody's suffering pretty bad today. Okay. I'll put you on breaks soon, unless you guys want a break right now. Okay. All right. So we've got our initial. We're talking about the initial entry of merozoites into red blood cells at this point. What happens? They get into the red blood cell, and first stage is a diagnostic value is this ring stage. The parasite gets into the red blood cell, and it has a little, looking at Vivax here. This is the entire parasite. The outer part of its membrane will stain purple with Giemsa. In the inside, you have a vacuole, a food vacuole basically, so that it is part of the parasite, it just doesn't stain, and then you have a tiny little red stained nucleus for that parasite. But not a lot of, this is a cartoon, not an actual, they're not actually looking at slides. These can be difficult to differentiate, particularly with Vivax and malariae and Ovale. But with falciparum, uniquely, you do get multiple rings in a single infected red blood cell. So that can be distinctive and diagnostic use. This isn't showing species I was mentioning, which also have multiple ring forms. So it looks very much like falciparum in this initial ring stage. Minimal damage to the red blood cell at this point, which is basically gone and entered. After entry, it's preparing to replicate. It's not quite replicating yet. Yeah, go ahead, Zach.
What's happening? Like it gets into the red blood cell as a merozoite, but then it's in the ring. What's happening between that and it leaves as a merozoite?
Yeah, it's a little bit confusing. So these are the developmental stages. It's not a ring; it's the ring stage. So the parasite is a merozoite. What's emerging when it lyses that red blood cell is a bunch of merozoites that are divided, but it's still merozoite. So think of the ring stage as nothing more than a diagnostic stage. It's just the parasite. So it is now inside that red blood cell. Remember, with red blood cells, these are not nucleated. There's not a lot of, there's no transcription or translation. The only purpose of red blood cells is to carry hemoglobin, right? So what the parasites are doing is they're destroying and metabolizing the hemoglobin in those red blood cells. As they break that down, that's going to start releasing this malarial pigment called hemazoin into the red blood cell. You can start seeing that in this trophozoite stage. So all this pigmentation you see out in these various species of Plasmodium, that's a breakdown products of hemoglobin. That is toxic. Because this stage is mostly feeding and metabolically active to asexually divide and replicate, you have this nevoid, purple staining parasite, various important species. The trophozoite stage is one of the easier stages to distinguish these species, particularly with malariae, Plasmodium malariae. Look at this one, it's got this distinctive band and bar trophozoite stage here. Ovale too. Ovale is a lot like Vivax in a lot of ways, but the unique thing about Ovale here is what it does to the erythrocyte of that point. Notice this roughed up, figurated edge of Ovale. That's just starting to do a lot of damage to that erythrocyte, causing distortions. At this point, those blood cells are not able to function as red blood cells. They're no longer carrying oxygen. Hemoglobin is broken down, so you start seeing some symptoms associated with depletion of hemoglobin in those red blood cells. Then we move on to the stage. So this is where those parasites are actually starting to undergo schizogony and divide. You can get up to 32 merozoites, depending on the species. Notice with Falciparum, how many merozoites are coming out of that mature schizon stage, or lots of them, about 32. Contrast that with malaria. This is when I said that it tends to have low levels of parasitemia. One of the reasons for the low levels of parasitemia is the fact that you don't get a lot of merozoites when you get 8. Another distinctive feature to watch out for with the schizon stage is this malaria pigment. It's the center for malaria, and a little bit for Ovale as well. It's sort of rosette merozoites around that malarial pigment in the center there. And then you don't see a stage after the schizon stage. What's happened to the red blood cell once it's gone? It's lysed, it's burst, and it's released all those merozoites. So, if you were lucky, you might see a relatively small number of red blood cells that have this differentiated gametocyte inside of them. Now they're diagnostically distinctive stage, particularly with Falciparum. Falciparum has this famous banana or crescent-shaped gametocyte. It's super easy to ID. The rest of them just have a bunch of nuclei, red staining nuclei that you may or may not be able to distinguish as easily. But again, these are the forms that will be picked up by the mosquito to transmit and then further propagate the life cycle. Okay, let's take a break there. We've got 10:43. Let's come back to 10:53 and pick up on Plasmodium.
Mary.
She might not be seen in our I think military provides new stuff like that. Because then
No, so.
I did say that. You're not saying the right stuff. It's on as a policy president. So if we just had another we have the same that's even just it has been helpful. Pick about 3 days. But all kind of like I took it about 2 days. Like average. At first, it feels like you use it like the first time. Your body's not used to it, like so I mean, for me, it's really down to my skin. I went out like
Okay, guys, let's drive on. Okay, so we're talking about the most deadly of the already pretty deadly Plasmodium species, Plasmodium. Going back to our maps, if you don't have that map in the beginning at all, that's fine. This is kind of important to know to look at this map. This is where most of those cases of falciparum occur. Here in sub-Saharan Africa, extending all the way to West Africa and Africa. And then notice that we've got cases that can happen in South America, northern South America, Brazil, Venezuela, cases that happen in India, Southeast Asia, into Malaysia, Indonesia, Australasia, etc. So pretty widespread, but concentrated heavily in sub-Saharan Africa. Responsible for 50% of all malaria cases worldwide. And again, the vast majority of severe or fatal malaria cases caused by this one. Talk about some of those reasons why. Fortunately, at this point, we have actually gone through a lot of material that's on this slide. I'll still try to go fairly slow so we don't charge through it, but it does specify shorter incubation periods. So I'm talking, when I say incubation period, I'm talking about from the time that you get the mosquito bite and the sporozoites are released into your blood and start honing in for your liver for that exoerythrocytic cycle, to the time you become symptomatic and start entering into those paroxysmal stages. In that period, you might get some flu-like symptoms, usually nothing too distinctive. Some people, especially when infected and adults, may clear the parasite and you don't progress to more severe diseases. Others will progress and they may fall into a quotidian, as in like everyday expecting a fever cycle, or eventually a tertian cycle. So you might start off quotidian and then shift to tertian later on as the disease progresses. We have that whole slide about signs and symptoms of severe malaria infections, complicated malaria. There's another word for it you might hear occasionally. It's called malignant tertian malaria. Malignant just referring to the fact that you can have a fulminant disease. Poor prognosis when it gets to be that bad. See hyperparasitemia. You guys remember what we find as hyperparasitemia? 5% of blood cells infected. You have patients where they have 10% of their blood cells infected. So you can get higher than 5%, but usually that correlates pretty well with severity. Diagnostics. So here's this nice blown-up example of what that Vinex Now RDT, the rapid diagnostic test, looks like for malaria. It detects a protein, HRP, I forget what that stands for, maybe it's HSP, but regardless, it has Plasmodium falciparum on there, and then it has PD, PM, PO as well. So it has a standard universal marker for these plasmodium species, as well as a secondary marker that gives you an idea if it's falciparum or some other species. It is really, really good for this species. These rapid diagnostic tests are good for falciparum, so you can remember that. Prevention, prophylactic medications. How many of you guys have been on prophylactics for malaria? Quite a few of you. Okay. So the big ones, you get Doxy. Each one of these has a downside, remember. Doxy has its own downside. So, yeah, the one associated with that's more psychoactive is methylphenidate. The nice thing about methylphenidate, if you don't have a history of psychiatric disorders, is it's taken on a weekly basis. So if you're going to be going to an endemic area for the long term, methylphenidate is a nice choice for that fact. I think it's otherwise fairly well tolerated. And then Malarone is the third one. Malarone is a daily, unfortunately, but that's very well tolerated if you can be someplace for a short period of time. But if you're living in one of those endemic areas, you're not going to be on prophylaxis probably, or if you're just there for a long time, it's just hard to stay on those. Because of that reason, if you're going to be in an endemic area, wearing bug spray, sleeping with mosquito nets, all those things become a huge nuisance after a while. Still very important to do that if you can. Vector, I do have a book to recommend to you guys. It's called Mosquito. It's a nonfiction book, Timothy Weingard. I forget when it was published, but super interesting book. Have you read it? Yeah? Okay. So, the entire second chapter, it talks about the biology of mosquitoes sucking blood and how sophisticated that process is. Like, and it's riveting. It's really, really interesting. And then the subsequent chapter about all the control measures that can be done historically, like the genetically modified mosquitoes. It talks about that. It talks about the horrible ecological things we did, the disastrous ecological things like draining swamplands. It talks about the effect it had on military campaigns. Super interesting book. And I'm putting together a book list for you guys, which I'm almost done with, and a music list. That's a really good book. Vaccine accident. These vaccines have been a long time coming. Follow this closely, or how they specifically prevent disease. But my understanding, particularly with mosquito rips, the first one, is that it stimulates the adaptive immune response. You get a humoral response that prevents those, like sporozoites, early in infection after you get bitten by a mosquito from binding to target cells. And then you also stimulate this cell-mediated immune response as well. So you get, particularly like CD8 T cells, will be more adept and more proficient at killing infected red blood cells and hepatocytes. So that's basically how those vaccines work. Really beneficial. I don't know how many people have been vaccinated or how available these vaccines are, but hopefully that continues to be something we can roll out and help bring down those numbers of those cases worldwide. I wish I had a laser pointer here. So, talking a little bit about what you might expect to see in a thin smear of a patient who has Plasmodium falciparum. Unfortunately, the slide has these in reverse order, and I've been too lazy to, cut and paste them, crop them, and put them in a logical sequence. But if you remember, the ring stage is the first stage you'd expect to see after entry of those parasites into a red blood cell. With falciparum, remember the things you're looking out for are multiple ring stages in a single red blood cell. That's considered to be pretty distinctive, as well as the appearance of these things hanging out to the outside of the cell. That's called applique or appellae. They just appear that way, though. They're not actually stuck on the exterior of the erythrocyte. But if you're one of those people that engages in arts and crafts, perhaps you're familiar with what applique is. It's really tacky craft But yeah, that's distinctive as well. That was a bit judgy, I'm sorry. So on this slide, keep in mind you do have trophozoite stages. That's the stage where these things are growing in size and metabolizing, breaking down hemoglobin. You definitely have schizon stages as well, when they're undergoing schizotony and producing huge numbers of merozoites. But since those are sequestered, you're not going to see them very often. You can still see pictures earlier on the deck if you're curious what that looks like, but just know that they're not here because of the fact that you are not likely to see them. And then the gametocytes, finally, this is the most distinctive thing about P-Fel7. You get that classic crescent banana-shaped gametocyte. Very easy if you're lucky enough to see it. These would not be something we see a whole lot of. Moving on to vivax. A couple interesting things about vivax. One is its distribution. This is the one where we still occasionally get cases of autochthonous in the United States. We get lots of import cases of vivax. And then just looking at the geographic distribution, that's what we're noting here. It's a vivax worldwide. If you notice, if you remember that slide when we were looking at Plasmodium falciparum and how the range extended all the way up to West Africa here, a lot of the West African population is not that status of vivax. That is because a lot of them are missing the Duffy antigen group on their red blood cells, so that makes them somewhat resistant to infection. That's the reason why most of the cases in Africa are across Africa. In terms of periodicity, vivax adheres to that 48-hour tertianity. It gets the title benign tertian malaria because the symptoms are indeed milder than falciparum. But remember, all 5 species can kill you, including this one. Another really, I guess, negative nature of vivax is that tendency to form hepatozoites in the liver. So early on in infection, before they progress to the erythrocytic cycle, those sporozoites can infect liver cells and basically just form quiescent hepatozoites. They're not actively replicating, they aren't attracting a lot of immune attention, and they can stay dormant there in the liver for months or years, and then spontaneously reactivate, and then you get vivax malaria at that point. So that's a significant cause of malaria morbidity associated with vivax, this delayed presentation. In terms of the thin smears for vivax, we are looking at a couple of unique features here. First is their tendency to infect young red blood cells. Have you guys talked about erythropoiesis at all in A&P? A little bit. Okay, so what's the average lifespan of a red blood cell?
2 months, 90 days.
About 6 months, about half a year. Yeah. And so when they're being produced in the bone marrow, it takes a little while for them to develop. Immature reticulocytes undergo circulation, finish developing over the course of a week or 2. But as they're circulating throughout the body, doing their thing, carrying the blood around, they continue to mature. They have their own arm. The young red blood cells are different than older red blood cells since they start aging out before they get taken out of circulation and destroyed by the spleen. Their membranes become increasingly rigid. So a young, immature red blood cell has a more flexible membrane, and that's the type of cell that these species of Plasmodium like to infect. Plasmodium vivax it favors the young, flexible ones, infecting those. And as it does that, it has a tendency to cause distortions in those infected red blood cells. It causes them to enlarge and become expanded. In the trophozoite stage here, you're looking at larger than normal red blood cells. It's that flexible membrane. Because they are restricting themselves to the age of the red blood cell, that limits the level of parasitemia. Falciparum has no age preference. It equal opportunity infects all age red blood cells. But when you narrow your population down to the young ones, you don't get high levels of parasitemia. Once you get to the schizon stage, about 24 parasitoids. I've never thought about them, but a more distinct feature might be that hemozoin pigment that sometimes you can see more in the center. You also see Schuffner dots, these pink cytoplasmic granules that are also breakdown products in cytoplasm. cartoon of it is here on the Binsner side. And then the commuter site stage, just very round, easily identified for that reason. Back to the next stage, only one ring, only one parasite per red blood cell, not multiple. Ovale, this is responsible for a relatively small percentage of all malaria cases, less than 5%. Western Central Africa, I don't have a map on this one, but it has a fairly restricted range, Western Pacific. In a lot of ways, it's similar to Vidax, but there are some unique differences as well. It has a very similar clinical presentation. It is also benign tertian malaria. It also is able to establish that dormant phase hypnozoites in liver cells and can cause relapse months or years down the road. Typically milder illness. But when you look at the thin smears, there are some differences that are notable. So the name Ovale, once you get to a trophozoite stage, it too has a preference for young viable red blood cells, but it causes a difference in distortion in those erythrocytes. It causes them to become ovoid in appearance. They're easy to remember: Ovale, ovoid, trophozoite stage, more distinctive. And remember on that card, I showed the rough fimbriated edges of the erythrocytes. That's a very distinctive, useful feature, particularly in the trophozoite stage. I don't know how many merozoites or viruses in that schizon stage. It looks like it's pretty packed, but distinctively you can see sometimes hemozoin, that pigment in the center of that schizon before the cells rupture. Malaria, I also don't have a map for Plasmodium malariae, but this is more co-endemic with a lot of falciparum cases. So you can just study that out if you're studying malaria. It has quartile periodicity, the longest and most distinctive of any of them, every third day, every 72 hours. Very long incubation period. It has mild and chronic infections. I'm sorry, this bullet is totally redundant. Bullet 2.5, but chronic mild infections generally. Low levels of parasitemia. And low levels of parasitemia associated with Plasmodium malariae is due to the fact that they too have a preference for particular age demographic of red blood cells. So they favor the old ones, the old, rigid, mature erythrocytes. And that keeps the levels of parasitemia low, and their options pretty restricted when infecting red blood cells. When those old red blood cells are infected, those rigid membranes prevent them from growing out and distorting the size of the red blood cells. So once they get to the trophozoite stage, when they're at their most metabolically active, preparing to divide into schizons, that's when you see this distinctive band and bar pattern is distinctive for Plasmodium malariae. That band and bar pattern is that trophozoite form, that amoeboid form that is just densely compacted into that erythrocyte. It has a membrane that is not giving, so it's very compact like that. Once you get to the schizon stage, you have relatively few merozoites, only have 8 of them, so you're getting huge numbers of infected merozoites at least when these red blood cells rupture. And that also contributes to the milder disease presentation with Plasmodium malariae. If you can see the xenon dots, that's distinctive for Plasmodium malariae. They're just reddish, like more reddish granules. That's considered to be a hallmark for Plasmodium malariae. And lastly, we've got Plasmodium knowlesii, or Knowlesi, who's named after a guy named Robert Knowles. So the pronunciations for this vary significantly. I say Knowlesi, but you'll hear it Knowlesi and some other pronunciations as well. This is a very interesting species. It's the latest and most recent one to be discussed. It's the one that I mentioned is a zoonotic, and that it is the primary reservoir of this mosquito attack. It's native to parts of Malaysia and neighboring countries in Southeast Asia. The mosquitoes that propagate Plasmodium knowlesii tend to have a unique gravitation towards the upper layers of the forest canopy, where people are not. So because of that reason, we don't have as much contact with the human population, and that has probably slowed the ability of this parasite to spill over into human populations. The key word there is spill. So this is maintained and adapted to a mosquito macaque lice cycle, but it's an example of a Plasmodium species that is slowly adapting to human populations. That has implications for our habitation and our deforestation in that part of the world, and coming in increasing contact with these parasites. I have another book plug for you guys. This book, it's on my list for you guys. It's called Spillover. Chapter 3 is about Plasmodium knowlesi and its identification and its characterization when it was initially just thought to be strictly disease maintained in primate populations to when the first human cases started to appear, and then they characterized it and found that kinetic is responsible for some pretty severe malaria cases. We talked about falciparum being the most deadly, the most dangerous, but when you're infected with this spillover zoonotic species, knowlesi, you tend to get really high levels of parasitemia. High parasitemia, severe disease, on par with falciparum. It doesn't have a cytoadherence though, so the pathology you talked about is Sigma 4 falciparum with the obstruction of microvasculature is absent from knowlesi, but it does affect red blood cells of all ages, so you do have high levels of parasitemia and some pretty severe disease. But this bullet here about neurological complications being there, that's because it doesn't have cytoadherence. It's because it doesn't have the clog up the blood vessels that are in the brain. What else here? I do not have a picture of the stages of knowlesi, but that's okay because they resemble other species. The ring stage looks like falciparum, so you can have multiple ring stages. The later stages look like Plasmodium malariae. Guess what that distinctive feature was in the trophozoite stage of Plasmodium malariae that we just talked about? Band, exactly. So that's what you get with Plasmodium knowlesi in that trophozoite stage. Easy to diagnose if you're only looking at big smears. So for that reason, clinicians have sort of a couple things in mind if they're trying to diagnose knowlesi malaria. High levels of parasitemia, which is usually either falciparum or knowlesi. Severe disease, which is usually either knowlesi or falciparum, and then recent travel to Malaysia or Thailand, Vietnam, Cambodia, or some of those regional areas. Those three together, you might start thinking of knowlesi in the absence of PCR or also in the absence of PCR. Okay, I'm debating whether or not to drive on with Babesia. You guys look like you're absolutely exposed. You are. Okay. Well, some people want to go on. Are you? How many people want to do Babesia and then holiday, or leave it all for tomorrow? How many people want to do Babesia and then holiday? How many people want to go home? Tiebreaker that looked even. Just kidding. I shouldn't put you on the spot like that. For sure. Okay, alright. I feel bad that it's really hot in here and you guys look miserable. Okay, let's just do pretty easy. Pretty easy. Similar a lot of ways. So we can get through it quickly. Okay, this is another blood protozoan. We're just talking about a lot of features of Plasmodium species. This is another member of P. complexa, so it still has the same body morphology, that corseted cytoskeletal arrangement. It still has the same a complex that allows it to penetrate into your blood cells, and it does infect red blood cells with Plasmodium. So in a lot of ways, Babesia, or babesiosis, that's the cause for Babesia, is similar to presentation with malaria. That'll be important as well. It is not transmitted by mosquitoes, it's transmitted by ticks, a couple other different modes of transmission dominant ticks. And the tick species, Ixodes, is one of the major tick species that can transmit Babesia. It's kind of hard to see with this tiny little font there, but the Ixodes In the United States, the type of Babesia we have on the species specific here is called Babesia microti, and it has heavy endemicity in areas like the northeastern part of the United States and New England, as well as the upper Midwest, Wisconsin, Minnesota. Basically, like the same areas that we talked about when we talked about Lyme disease, Borrelia burgdorferi, same areas again, beyond babesiosis. So it has a hot spot in areas in like Nantucket and Massachusetts, Martha's Vineyard, lots of cases of babesiosis there, and most of those are transmitted by tick bites. If you go over to Europe, there's a different species but a similar presentation. That's Babesia divergens. Tends to be a little bit more severe, but in this map you can see Babesia divergens and some other species as well across the globe, but those are the two that we're focusing on. Other notable transmission modes would be transfusion-related babesiosis, TB, transfusion-transmitted babesiosis. And that is because we have poor screening for Babesia in our blood supply. If you have ever tested positive for babesiosis, you get put on indefinite deferral, so you basically never donate blood again. But there is no FDA history test in our blood supply. So you do have cases of transfusion-transmitted babesiosis. And there's a hidden slide in your deck if you want to learn a little bit more about that, but it's not going to be testable here. For prevention, it's basically the same things that you would do to prevent Lyme disease, removing those ticks, scan, doing tick checks to make sure that you're aware that you've been bitten by a tick, wearing repellents. And this last bullet, asplenic individuals. If you know you're asplenic, there are actual travel warnings not to go into areas that are hyperendemic for babesiosis. Why would having a spleen matter for cases of babesiosis, do you think? Why is it, I mean, it's important for a lot of people, particularly why for babesiosis. Yeah, we talked about the function of the spleen before. It's filtering out the blood, right? So all these erythrocytes that are infected with blood parasites like Babesia or like Plasmodium, you need a spleen in order to filter those out and destroy them. Basically, without a spleen, the disease is much more severe and the prognosis is much poorer. In terms of the life cycle, good news, it is much easier, I think, than the life cycle of malaria. You get bitten by a tick, there's a significant latch required to transmit this infective form, which is these pyriform bodies, sporozoites. They get into your bloodstream. They do not go into the liver, they go straight into infecting erythrocytes, and then they have a relatively simple replication cycle. They reproduce through binary fission, asexual reproduction of the human host again. They get amplified in these infected cells. Populations get amplified. That will contribute to the primary clinical features, which is fever, and we'll talk a little bit more about that in the next slide. In terms of diagnosis, we'll also talk about that, but there's a distinctive form where my laser pointer is here, where you have the parasites divided into four different organisms, basically the same infected red blood cells. Before it ruptures, it forms what's called a Maltese cross. Let's see a picture of that. But as much as this parasite looks like Plasmodium, if you see a Maltese cross, a tetrad of Babesia, an infected red blood cell, that's considered to be diagnostic and distinctive for Babesia. So that's important. When you get bitten by a tick, that's what they're taking off the infected form. And then the ticks too serve as a definitive host. So sexual reproduction of the parasite will occur in that tick species. Uniquely, this is something that doesn't happen in mosquitoes, to my knowledge, but in tick populations, in order to propagate these parasites in the tick populations, they can transmit the parasite from mother to offspring. That's called transvariant transmission, and that keeps the parasiticity in the tick population fairly high. It allows it to circulate better. Clinical features. We've got a prodromal phase, and then some pretty generic symptoms that could look a lot like malaria, but without the periodicity, basically. So when you get fevers, they do also correspond to the rupture of red blood cells, but the red blood cell ruptures are not synchronized like they are with malaria. So the fever cycles are much less predictable. But all the other symptoms of headache, chills, sweating, fatigue, weakness, that's all pretty much the same. And a similar pathology, you get hemolytic anemia when the blood cells are destroyed in large numbers. It's extreme weakness. If this gets really bad, you may start having renal failure, kidney failure. Your liver too can be impacted in really severe disease. In terms of diagnosis, at the bottom here we can see this is the Babesia, but it looks so much like malaria, doesn't it? So the problem here, I've heard ID docs lament the fact that microscopists are undertrained and inexperienced and unable to differentiate between these two diseases. But that is the issue. If you're relying solely on thin smears to diagnose, very easy to confuse those. It is possible to distinguish them from thin smears alone, especially if you find that distinctive Maltese cross. It's like in the top here. That was top stuff. 4 different organs, and they're arranged in a very distinctive way there. You don't always find that though, so that's the challenge. Hopefully you're in a place that can do PCR and you can get a definitive diagnosis that way, or you can do serology. You can look for IgM for acute infection, diagnose from that. But it should be in the differential diagnosis if you're in one of those parts of the country where Babesia is somewhat endemic into the population. Okay. All right, guys. I will let you go. I'm so ready to drive on with toxoplasma, but I think we should just call it. Yeah, sorry. We'll do it tomorrow. I swear, if you guys could see what I see when I look out there, you would stop talking to me.